Hello, my name is Isabella de la Paz, and today I will be presenting based on Krimmel and Pierce's scientific paper published in 2013, Sticky Plants Trap Insects to Enhance Indirect Defense. The main objective of this study was to determine whether sticky plants that trap insects can indirectly defend themselves by using the trapped insects or insect carrion as food for predatory arthropods. Specifically, the study tested if this carrion-based food source increases predator populations, reduces herbivory, and ultimately enhances plant reproductive success. Our first research question being, does adding insect carrion, in this case dead Drosophila fruit flies, to sticky tarweed, specifically Medea elegans plants, increase the abundance and diversity of predatory arthropods? The second question, does the presence of more predators on carrion augmented plants lead to lower herbivore damage, particularly from the caterpillar Heliothodes diminutiva? And the third being, do plants that trap or receive added carrion show higher fitness, measured by the number of fruits produced compared to the control plants? The first hypothesis, entrapped insect carrion serves as a plant-provided food source that attracts more predators, especially species adapted to sticky surfaces. The second hypothesis, increased predator activity from carrion addition leads to less herbivore damage by the caterpillar. And the third hypothesis, reduced herbivory increases plant lifetime fitness, demonstrated by higher fruit production. The broader expectation that the researchers had with this study was that sticky plants benefit from a mutualistic interaction. Their glandular trichomes not only deter herbivores directly, but also support predators indirectly by offering carrion as a protein-rich resource. And here is a flowchart that I made based on the researchers' expectations of this study and their hypothesis. Some background on this study. Plants use both direct and indirect defenses to protect themselves from herbivores. While direct defenses, like chemicals, physically deter or harm herbivores, indirect defenses rely on attracting predators and parasites that attack the herbivores. Common plant-provided foods that help attract these beneficial predators include pollen, extrafloral nectar, and protein bodies, which supply nutrients and encourage predator presence. The knowledge gap of this study is that recent research has proposed that insect carrion, which is typically dead animal carcasses, but in this case it's dead insects, could also serve as a valuable food source for predatory arthropods. But this idea had not been experimentally tested in a natural system before this study. Sticky plants that trap small insects with glandular trichomes may unintentionally provide this carrion, creating a new kind of plant-provided resource that could strengthen indirect defenses. This study investigated and focused on whether entrapped insect carrion on the sticky plant, Medea elegans or common tarweed, can enhance indirect plant defense by supporting predators that reduce herbivory. The researchers hypothesized that sticky plants benefit not only by directly deterring herbivores, but also by indirectly defending themselves by using the trapped insects as a protein-rich food source for predatory arthropods such as assassin bugs, stilt bugs, and some spiders. The importance of this study. Understanding this mechanism reveals a previously overlooked ecological interaction where plant stickiness may benefit both the plant and its predators, rather than simply deterring insects. This work challenges the traditional idea that sticky trichomes are purely defensive and shows how they can foster mutualistic relationships that improve plant survival and reproduction in nature. And here we have a picture from the actual scientific paper itself of the tarweed and of some of the predator bugs in action. So this study was conducted at the Stebbins Cold Canyon Nature Reserve in Solano County, California. I have it marked there on the map. It is an oak, charple savanna kind of ecosystem, and the research focused on Medea elegans, the common tarweed, which is a native annual plant that's covered in glandular trichomes, which makes its surface sticky. The main herbivore studied was the caterpillar, Heliothodes diminutiva, which feeds on the plant's buds and flowers, while common predators included assassin bugs, stilt bugs, and lynx and crab spiders, all capable of moving across sticky surfaces. For the experiment, two complementary field experiments were conducted to test whether entrapped insect carrion enhances indirect plant defense, the first one being the carrion addition experiment. The setup was 82 tarweed plants were randomly assigned to either control, which is no carrion added, or carrion added, which had five freeze-killed Drosophila fruit flies placed weekly on the plant's trichomes. It lasted from early June through September, covering the plant's entire growing and reproductive season. Each week, researchers recorded the number of predators, herbivore damage, which was the chewed buds, naturally trapped carrion, and fruit production as a measure of the plant's fitness. They also ranked plant stickiness, trichome density, and secretion activity on a scale of 0 to 5. For the second part of the experiment, which was the caterpillar addition, the setup was 14 pairs of additional plants were selected. One plant in each pair received a live caterpillar, while the other served as a control without a caterpillar. The purpose was to measure the direct effect of herbivory on plant fitness, and the measurements included weekly observations of bud damage and fruit production, which quantified how herbivory reduced reproductive success. The researchers condensed weekly data into season-long averages for each plant to evaluate treatment effects. They compared predator abundance, herbivore damage, and fruit yield between the carrion added, caterpillar added, and control plants. This design allowed them to isolate the effect of carrion availability on predator recruitment and indirect defense effectiveness. And here we have a flowchart from the actual scientific paper, which includes their expectations for their experimental design. So, first we'll start with predator abundance increased with carrion. We're on to the results portion. So, adding 5 dead Drosophila flies weekly to Medea elegans plants resulted in a 76 to 450% increase in predator abundance compared to control plants. All 5 major predator species, assassin bugs, stilt bugs, lynx spiders, and crab spiders, were found in greater numbers on carrion treated plants. This effect was strongest in the season June to July when predators were colonizing new plants. Reduced herbivory from the caterpillars. So the carrion-enriched plants experienced a 60% decrease in bud damage caused by the caterpillars, the dominant herbivore that feeds on flower buds. This reduction in damage shows that the increase in predators effectively enhanced top-down control of herbivory. Conversely, when the caterpillars were experimentally added in a separate test, those plants suffered 50% greater bud damage and produced 50% fewer fruits, confirming that herbivory directly reduces plant reproductive success. Higher fruit production and plant fitness. Plants with added carrion produced about 10% more dehisced fruits than control plants, indicating a measurable improvement in reproductive success or fitness because M. elegans reproduces only through seed production. This fruit increase represents a significant gain in lifetime fitness. Trichome stickiness and predator activity. Plants with denser glandular trichomes or stickier surfaces entrapped more insect carrion overall. In control plants, without the carrion, those that naturally trap more insects also had a higher number of assassin bug eggs, showing that natural stickiness alone attracts predators and supports their reproduction. Furthermore, with the results, here we have diagram 1, which is the seasonal trends in predator eggs and herbivore damage. In the first graph, we see how adding insect carrion, specifically the dead Drosophila flies, to the Medea elegans plants influenced the ecosystem over the growing season. The top panel A shows that assassin bugs laid far more eggs on carrion-treated plants than on control plants, especially early in the season around June and July. This means that the presence of carrion quickly attracted predators and boosted their reproduction. In panel B, we see that opposite pattern with herbivory. As carrion and predators increased, bud damage from the caterpillars stayed much lower through most of the season. So this figure tells us that trapped insect carrion helped recruit beneficial predators early on, which helped the success of herbivores later in which helped then suppress herbivores later in the season. For the second diagram, this figure compares the control and carry-on treated plants directly. In part A, you can see that all predator species, assassin bugs, stilt bugs, and spiders, were significantly more abundant on plants with added carrion. Some increases were dramatic, up to several times higher than on the control plants. The stars on the bar indicate strong statistical significance, meaning these differences are real and not random. Then part B shows the downstream effect. Carrion treated plants had fewer damaged buds and produced more fruits. This means that the added carrion not only boosted predator populations, but also led to less herbivory and higher reproductive success. Clear evidence of an indirect defense mechanism benefiting the plant. And then finally, this third figure focuses on the caterpillar addition experiment. Here, one caterpillar was added to each experimental plant to test how much herbivory alone could impact fitness. As shown in panel A, plants with caterpillars suffered much higher bud damage, about 50% more than control plants. In panel B, those same plants produced about half as many fruits. This result confirms how destructive herbivory is for Emma ligands, reinforcing that predator protection, like that provided by carrion attraction, is crucial for the plant's survival and reproduction. Together, these results show the full story. Carrion attracts predators, predators reduce herbivory, and plants ultimately produce more seeds and more fruits and thrive better in the ecosystem. To conclude, this study concludes that entrapment of insect carrion by sticky plants functions as an indirect plant defense mechanism. By trapping insects on their glandular trichomes, Medea elegans provides a supplemental food resource for a community of predatory arthropods, such as assassin bugs, stilt bugs, lynx, and crab spiders. This carry-on provisioning sufficiently increased predator abundance by 76% to 450% and reduced herbivore damage by 60%, leading to a 10% rise in fruit production and overall plant fitness. These findings demonstrate that sticky trichomes are not merely physical defenses, but also ecological tools that enhance top-down control of herbivores. The broader importance of these results and this study lies in showing that indirect defenses can arise from scavenging interactions rather than only mutualistic nectar or protein body systems. Because carrion provides a protein-rich resource without direct energetic cost to the plant, this mechanism helps explain why glandular and hooked trichomes are so widespread across flowering plants. It challenges the traditional view that sticky trichomes are costly due to predator interference and instead suggests that they may create an ecologically efficient trap system that benefits both plants and predator species. This discovery broadens our understanding of plant arthropod mutualisms and highlights how even accidental insect entrapment can contribute to ecosystem balance and evolutionary success. And here I have my reference, which is our scientific paper and study. Thank you very much for listening to my presentation.